How are CFTR mutations identified from cystic fibrosis genetic tests? Imagine unlocking a secret code in your DNA that can tell you if you carry or have cystic fibrosis. That's what genetic testing for the CFTR gene does. The CFTR gene makes a protein that helps control salt and water movement in your cells. When there are mutations in both copies of this gene, it can cause cystic fibrosis. To find these mutations, scientists analyze your DNA, which they usually get from a blood or saliva sample. The first step often involves targeted mutation panels. These panels look for the most common mutations in the CFTR gene. For example, the American College of Medical Genetics recommends testing for 23 common mutations, including the well-known delta F508 mutation. These panels are designed to catch mutations that are most frequent in the population and are useful for initial screening of carriers or diagnosing someone with symptoms. If the initial test doesn't find any mutations but suspicion remains high, the next step might be expanded mutation analysis. This involves genotyping arrays or targeted sequencing that can detect hundreds of different CFTR variants. This broader approach increases the chances of identifying less common or rare mutations that could still cause cystic fibrosis or related conditions. When these tests still don't reveal any mutations but clinical signs suggest cystic fibrosis, doctors may recommend full gene sequencing. This process reads every part of the CFTR gene, including all the exons or coding regions, introns or non-coding regions, and regulatory areas that control gene activity. It also looks for large deletions or duplications that might not be caught by sequencing alone. These structural changes can be significant, so comprehensive analysis ensures nothing is missed. Once mutations are identified, experts interpret what they mean. They classify each variant as disease-causing, benign, or uncertain. This interpretation often involves comparing the variant to known databases, family studies, or functional tests to see if the mutation affects the CFTR protein's function. Sometimes, variants of uncertain significance requiring further investigation to understand their impact. Specialized programs, such as those run by the Cystic Fibrosis Foundation, offer advanced testing options. These include full sequencing and deletion or duplication analysis, along with expert interpretation to help clarify complex results. Reflex testing may also be performed for specific variants, like the 5T variant, especially when other mutations like R117H are found, to better understand how the mutation might influence disease severity. Throughout this process, genetic testing results are considered alongside clinical findings like sweat chloride tests and symptoms. Genetic counseling plays a vital role in explaining what the results mean for the individual and their family. It helps assess carrier status, reproductive risks, and potential eligibility for treatments that target CFTR mutations. In summary, identifying CFTR mutations involves a step-by-step approach, starting with common mutation panels, expanding to comprehensive gene sequencing and structural analysis when needed, and always being carefully interpreted by specialists. This thorough process helps ensure accurate diagnosis, guides treatment options, and provides valuable information for families. Always remember, for personalized advice and understanding your specific situation, consulting healthcare professionals is essential.